In this video, we're going to talk about molecular genetics, DNA, RNA, protein. We'll first start with DNA. What is the full name of DNA? The full name of DNA is called deoxyribonucleic acid. This name is really important because DNA is an example of the polymer called nucleic acid. RNA is the other example of nucleic acid. And the first part of DNA is called deoxyribo. So this tells you that the sugar that's contained within one molecule of DNA is called deoxyribose. So this sugar right here is called deoxyribose. The function of DNA is it stores and passes down genetic information. That's the function of DNA. What is the name of the type of sugar? We did that. Are all DNAs or genes the same size? The answer is no. So right here, it shows you DNA. This structure right here is a monomer of DNA. That's one DNA nucleotide, so we call it a nucleotide. There are three parts to it. This is a phosphate group, this is the deoxyribose, the sugar part, and this is the nitrogen base, or you can call it the base. There are four types of bases, A, T, C, and G, and C matches with G. The way I remember it is that C and G kind of look similar, and then A matches with T because those two are the leftover ones. What else do you need to know about DNA? DNA is double-stranded, as you can see over here, and is a twisted ladder or double helix. And it's also antiparallel, which means, as you can see over here, one side, the phosphate groups are pointing upward, and these phosphate groups are pointing down, right? So we call this antiparallel. Next question is, where is DNA stored? It's stored in the nucleus. Four types of bases, we just said that, A, T, C, G. They're called adenine, thymine, guanine, and cytosine. That's for DNA. Which base pairs with which base? We just said that. Is DNA double-stranded or single-stranded? Double. You can label it now. Okay. Next one is DNA is antiparallel. We already talked about that. You need to know that RNA, instead of having a T, so DNA has ATCG, RNA would have AU, C, and G. Okay? So instead of A matching with a T in RNA, if you go from DNA to RNA, you would have A matching with U. So what it means is that, let's say, normally you have a DNA of AAA, CTG. If you have another strand of DNA, it will be TTT, GAC. However, if you match the first strand into an RNA, it will be UUU and then GAC. Okay, see the difference? So right here we have the RNA matching with this DNA. If we're doing transcription, this is the result that you'll get. But if you're doing DNA replication, this is the result that you get from this strand right here. Next one is, what is the name of an RNA? It's called ribonucleic acid. So instead of deoxyribonucleic acid, you get rid of the deoxy part. The reason for that is RNA sugar has one more oxygen than the DNA. So DNA is called deoxy. It's missing an oxygen. The three differences between DNA and RNA are RNA has a ribose as the sugar. DNA is called deoxyribose. RNA has a uracil base, but DNA has a thymine base. RNA is single-stranded, so instead of having two strands, RNA only has one of those strands, and then DNA is double-stranded. What are some similarities? DNA and RNA are both nucleic acids. They both have AGC bases. They both have phosphate groups. So those are the similarities. The next one, what is the model process of DNA replication? We call this semi-conservative. So if you just look at this picture right here, during the semi-conservative DNA replication model, we start with one DNA. During the first replication, we end up with two DNA. But as you can see, with the color, there is one old strand, the blue one, and one newly synthesized strand. So it's kind of like ripping this original DNA apart into two separate strands, and then you add new DNA nucleotides onto here. Remember, a nucleotide is the DNA monomer. The next one is So the other two, conservative and dispersive, were models that people predicted and were then found out as incorrect. The next one is, which phase of the cell cycle does DNA replication occur? The cell cycle has 3 phases, G1, S, G2, M. Remember, M is the mitotic phase where the cell actually splits apart and replicate. But in order to prepare for replication, we have to have the S phase, the synthesis phase. That's when your little one strand is turned into an X shape. Remember, this is one chromosome. This is still one chromosome, but during the S phase, this one chromosome, or this one strand of DNA, is duplicated into 2 same strands of DNA. Okay. The next one is, what is the enzyme that allows nucleotides to bind to an old strand of DNA in order to allow the synthesis of the new strand? This molecule is called DNA polymerase. You just have to remember it. But it's kind of easy to remember because what you're trying to do is you're making a polymer of DNA, right? Instead of one nucleotide. A nucleotide is this one little unit. But instead of making one nucleotide, you're making polymer of DNA, one long strand of DNA. So we call this DNA polymerase. That's the enzyme. If you look at this picture, here's the DNA polymerase adding all those nucleotides onto the old strand. And we're forming the new strand. And eventually we'll have 2 separate DNAs. The other one that you have to know is that before DNA is replicated, the DNA has to be unzipped, right? Instead of having our winded up DNA, we have to separate it into 2 separate strands. What is the function of a telomere? So a telomere helps making sure that the DNA is complete, because every time we do a DNA replication process, we lose a little bit at the end. But if we add a bunch of sequences at the end of a DNA, that doesn't really mean anything, then this shortening of the DNA is not as big of a deal because it's not actually a part of your gene. Next question is, what is the definition of a gene? We already talked about that yesterday. The definition of a gene is a sequence of DNA that codes for a protein or a trait. What is the monomer of a protein? Amino acid. Polymer of a protein is called polypeptide. So when we look at an amino acid chart, what you see on it, such as this one over here, each one of these is an amino acid, okay? So that's an amino acid codon chart. If we add a bunch of amino acids together and form a polymer, you form a polypeptide. And then back to DNA one more time. Monomer of DNA is called a nucleotide. Monomer of RNA is also called a nucleotide. A polymer is called a nucleic acid. Can DNA be made into traits directly? No. Everything that you see of a person, everything that you can see, are expressed as protein. All right, so your eye color, those are proteins doing eye color things. You can't just see the DNA. So DNA cannot be translated into traits directly. How does it go through, how do you get from DNA to a trait? That's where we have the central dogma right here. DNA to RNA to protein. DNA to RNA is called transcription. This is something that's worth writing down, okay? Write down DNA to RNA, transcription. RNA to protein is translation. What type of macromolecule allow traits to be expressed? Like I said, proteins. Which organelle synthesizes protein? Which organelle? It would be ribosome. So there's an organelle called ribosome that makes proteins, right? Can DNA leave the nucleus? No, that is why we have this RNA, specifically mRNA, messenger RNA, to turn the DNA into protein, because the DNA cannot come out of the nucleus. So if you look at this picture right here, we have a DNA right here. A DNA is made into RNA, so now we have an RNA. This RNA now comes out of the nucleus, and then we have tRNA coming to the mRNA at the ribosome. So right here is not labeled, it's a ribosome. The ribosome allows you to make mRNA, right here, mRNA, into a protein. There's another type of RNA called rRNA. That's just a part of the ribosome that helps you with the process of making proteins. All right, mRNA is synthesized in the nucleus. It needs to move to the cytoplasm, as you can see in this picture. What is the central dogma? Like I said, DNA to RNA to protein, transcription, translation, we already talked about that. How many RNA bases are in one codon? So a codon is a sequence of RNA that can code for a specific amino acid. Remember, amino acid is the monomer or polymer of a protein. It's a monomer. The polymer of a protein is called a polypeptide. So there are 3 RNA bases, 3 nucleotide bases that can code for one protein. Well, one amino acid, one amino acid, 3 nucleotide, 1 amino acid. A bunch of amino acid, that's called a polypeptide or a protein. So one codon, 1 amino acid, like this person said. If you look right here, right, if this has to be an RNA, how do you know it? Because there are U's in it. DNA doesn't have a U. So this is an RNA. For each 3 bases of an RNA, you can make 1 protein, another protein, another protein. We'll do a practice in a little bit as well. 3 types of RNA. You can just take a look at this. I already talked about it. You need to know how to write a complementary strand of DNA, RNA, and protein. So let's do this as a practice. Write down these and figure out what the what the DNA replication other strand is, what the RNA is, and then what the protein is. So pause the video and come back. All right, now you're back. There's DNA. Our DNA other strand is going to be A matches with T, T matches with A, and then A again. G, C, C, and then T, G, A. If we're going to make an RNA out of the first strand, how's that going to work? A matches with U, T matches with A, and then G, C, C, and then A matches with U again, C, and then G, T matches with A. We're matching this strand with this strand. This is our RNA. Now, how do we know what kind of protein or what 3 amino acids are we going to produce, right? Remember, 3 nucleotides give you 1 amino acid. So if you were to have 5 amino acids, how many nucleotides do you need? That's going to be 5 times 3, which is 15. How many codons do you need? That's still 5, because each codon codes for 1 amino acid. All right, so now we're ready for protein. UAA. If you look in this chart, U first base, second base, look this way, UA. Now you find UAA is leucine, so LEU, and then GCC. You find on the left side G, second base, look to the right, C, GCC right here, alanine, ALA. And then we have the last one, UGA. U on the left side, G over here, and UGA. Stop. This works perfectly. So as you can see in this chart, there are some stop codons. Right? There's a stop codon right here. Oh, that's the only one. So what the stop codon, oh, there's another one right here. So what the stop codon does is that we are coding from mRNA to a protein. And at some point, you need to stop. The stop codon tells the translation process to stop. That's exactly what it means. All right, next one. What is a mutation? What are some causes? The mutation is a change in the DNA sequence or basis, or changing the DNA. However, since DNA is what codes for the mRNA and the protein, a change in the base of a DNA could have some very serious consequences. So what can happen? What causes this? Mistakes during transcription or translation, or as well as during DNA replication. So during DNA replication, transcription, and translation. But this one is the big one because a change in DNA sequence is a mutation. So the cause can be DNA replication. And then the next one, mutations in somatic cell versus gametes. So if you remember, well, you have to remember it, somatic cell is your body cell. Gametes are sperms and eggs. So everything except sperms and eggs are called somatic cells. Which one can be passed down to the offspring? So if you were to have a mutation in your somatic cell, let's say a mutation on your arm, you got a little tumor, is that going to be passed down to your offsprings? No, because it's not in the gamete. The only thing that you give to your offspring is the gamete right here. So in order for a mutation to be passed down to an offspring, it has to be a mutation within the gamete genes. Okay. Next one, explain point mutation and substitution. So a point mutation is the change in one nucleotide. So if we look right here, if we change this T to a G, that will be a point mutation. There are three possible consequences of a point mutation. There's a silent mutation, where even though you changed one base, let's say you changed this one, that's the original code, you change it to this. If the result is there's no change in amino acid, right? If you started with an alanine, but then after the one nucleotide change, you still get an alanine. We call that a silent mutation because there's no real consequences. There's no change in amino acid. Next one is called a missense mutation, where you actually changes the original amino acid to a different one. So let's say you're supposed to have an alanine at a certain point, but instead you got a valine. We will call that a missense mutation. What is a nonsense mutation is when originally you should have an actual amino acid that codes for something, let's say glycine. However, instead of the glycine, you ended up with a stop codon. That's when we call a nonsense mutation, okay? And the result is you have a premature stop codon, so your polypeptide chain protein is going to be shortened because you stopped before you're supposed to stop. The next one is chromosomal alteration. Has a frameshift mutation. If you insert a code, let's say right here, instead of this code, you add an extra one. Or if you delete one, instead of this one, you get rid of this C. We call that a frameshift mutation because when you're reading this code, you read TAA, CTG, right? But after you add this C, you're going to read TAA, CCT. Now your reading frame is shifted toward this way. That's why we call it a frameshift. Duplication is where you duplicate a part of the chromosome. Inversion is if you take a part, let's say you take B, C, D, E, and then turn it 180 degree backward, then this is what you're going to have. And the last one is called translocation, where you have two chromosomes, and then see right here, two chromosomes, and then you switch apart. So it's kind of like crossing over actually. Karyotype, these are just basic things that you should already know if you watched the video from last night. The normal amount of chromosomes that a human somatic cell has. A somatic cell is your body cell. It's diploid, and it has 46 chromosomes. Oops. And then so you should know that. Gametes are haploid. It has it's egg and sperms. It has 23 chromosomes in a normal human body. So what is the diploid number? 46. Haploid, 23. How many autosomes and sex chromosomes are in each somatic cells? Autosomes is going to be 44 pairs, 22 pairs or 44 individual chromosomes. And the sex chromosomes, you have 2 of those. So if you look in this picture, there are 44 pairs of autosomes. Autosomes is anything besides the sex chromosomes that determines your gender. And then we have X and Y. XY is male, XX is female. What happens if you have a non-disjunction? A non-disjunction will give you an abnormal amount of chromosomes within the daughter cell. So let's say we have meiosis, but the chromosomes didn't split as the way it's supposed to, and you'll have an abnormal amount of chromosomes. So for example, Down syndrome is when you have 3 chromosomes instead of 2. It's called trisomy 21. So you will have 3 chromosomes right here instead of 2. Next one. Oh, we're almost done. What is genetic engineering? It's when you're modifying the genetic material in order to change something within an organism. That's genetic engineering. The next one is right here. How is it done? You would do something called gene splicing. You take a DNA and you splice it, and then you replace it with something else. Okay, so that's gene splicing. You literally cut the gene open. but not with scissors. You have enzymes that can do that. And the next one is called, what is a recombinant DNA? This result where you have some DNA of the original, if it's a human, for example, and then you add a piece of bacterial DNA for some reason, that's a recombinant DNA. What is the function of a restriction enzyme? Restriction enzyme is the enzyme that actually does the cutting. Restriction enzymes are the scissors. What is the purpose of gel electrophoresis? Let's say you have a really long DNA and you cut it into 20 pieces. How are you supposed to know which piece is which? The first thing that you can do is to use gel electrophoresis and separate those pieces by their sizes. All right, now that you've watched this video, you're going to do really well on tomorrow's quiz. And I'm very happy that you're watching this video because it's supposed to be helpful. Bye.